Well, shall we get started for the morning? First, I'd like to welcome everyone. Thank you for coming to our NP seminar series sponsored by the FES Center and the APT Center. And this morning, we're really delighted to hold this in conjunction with the Department of Neurology here at the School of Medicine. Are there any announcements this morning? I have one announcement. Our next seminar speaker will be Pedram Mosheni, April 17th, 8:30 here. Professor Mosheni's interests are in the field of biomicrosystems, so it should be a very interesting presentation. This morning, I'm really delighted to be able to introduce Dr. Lara Boyd. She is a neuroscientist, and in her 7 short years since completing her Ph.D., she's published over 30 manuscripts, had 7 funded studies, numerous scholarships and honors along the way. She's already mentored 5 Ph.D. and graduate students. She is with the School of Rehabilitation Sciences and is director of her own research laboratory in the Brain Research Center at the University of British Columbia. She has as the past director of a Ph.D. program, educational program in rehabilitation sciences. Please come on in and grab a bite to eat. She has been the former director of the Ph.D. program in rehabilitation sciences at the University of Kansas Medical Center, and currently is the Canada Research Chair of the Neurobiology of Motor Learning at the University of British Columbia. Dr. Boyd.
Thanks. Thank you, Dr. Daly. Thanks, all of you. I understand it's a holiday, so I guess double thanks for actually coming out early in the morning. And I know it's hard sometimes to mobilize on spring break if you have other family commitments, so I doubly thank you. So this morning, I have a couple things that I was hoping to cover with respect to the work that we have done and that we're continuing to do in my lab. And the first is I want to just kind of paint a picture for everyone about what we're learning with respect to how the stroke-affected brain operationalizes learning. And in particular, just to kind of give a summary of some of the work that we've done, I've tried to organize things around factors that we seem to be arriving at some consensus about that impair learning or slow learning, and then those that seem to facilitate learning in the stroke-affected brain. And then I'm going to transition to the second part of my talk and show you some of our new data and some of our ongoing studies where we're using transcranial magnetic stimulation to manipulate the brain and also to map the brain in association with motor learning. And this work is actually both in healthy control populations and then also into stroke. So just to kind of frame the talk today, and I know that all of you are well aware of this fact, but it really kind of is the organizing principle for what I do, is, you know, I'm always thinking about as we learn new data that our brains are reorganizing themselves at some level, whether it's cellular or at a systems level. You know, anytime we encode any piece of new information, we've essentially remodeled our brains in some fashion. And I would propose to those of you in the audience that this is really a theory of rehabilitation or recovery. So any rehabilitation intervention or recovery intervention that's successful is capitalizing on some kind of stimulant of positive neuroplastic change. And in the work that I do, it's always in a behavioral arena. So when we think about neuroplasticity and recovery from stroke, there's certainly an emerging picture and consensus that the central nervous system is highly influenced by behavior. And that, you know, as we all know, and it's a little unfortunate, but there's no magic drug we can take that will stimulate neuroplastic change or recovery after any kind of central nervous system lesion. Our direct cortical interventions, so cortical stimulation, deep brain stimulation, they help, but they're not a magic bullet. What seems to be the most effective kind of prerequisite for behavioral change or for neuroplastic change is behavior and repetitive behaviors. And when it comes to the motor systems, a lot of repetitive behavior. So kind of the purpose of my lab then is to understand which factors stimulate plasticity and to use this then to try to facilitate the development of new therapeutic interventions. There's some, I know you guys don't want them, but there's seats in the front. It's okay. Okay, so, and you're all well aware of this as well, I'm sure, that stroke is continuing to be a huge problem despite our best efforts. I'm an American, but now that I live in Canada, I have to incorporate my Canadian statistics into everything. The picture up there isn't any better than down here. So combined US and Canada, someone's having a stroke every 45 seconds. So I don't anticipate being put out of business anytime too soon, although that would be lovely. And one of the other things that we're particularly interested in my lab is trying to understand what happens to the more severely affected stroke patient. So we know a lot in the last couple of years about recovery in more mildly affected individuals. So things like the constraint-induced therapy trials that have gone on have showed us that these people are very able to recover when given the appropriate dose of rehabilitation practice. We know much less about more moderately and severely affected patients. And so that's something else that we're trying to think about very carefully. And I know that many of you are doing the same. So we have some elegant models that explain to us how recovery is operationalized after stroke. So this is now the really seminal paper by Randy Nudo. And these are essentially showing that when a focal infarct is induced into the motor area, the hand motor area of a squirrel monkey by an ischemic electrocoagulation technique, you produce a focal lesion. And that the recovery of the animal after these focal lesions is critically mediated by the behavior that that animal then engages in. So if you have nonspecific arm use and allow the use of the non-hemiparetic arm after a focal lesion, you essentially see a shrinking of the cortical territory devoted to the hand, the digit, and the forearm, and that's what's shown here. So this is a change in percent area of motor cortex. If you force the use of the affected arm with high dose and task-specific training, you see an actual enlargement of cortex and of adjacent cortex. So specifically, more enlargements in the hand and the wrist and forearm. So this information has really critically shaped the way we think about rehabilitation. And one key piece here in the development of these forced use therapies, whether you're working in either of these primate models, the key ingredient to each of these outcomes appears to be a very high dose, if you will, or a high number of repetitions in order to stimulate positive neuroplastic change in the brain. So in order to not lose territory in motor cortex that's devoted to hand and wrist. So if you look across the literature, and we've done this in a couple different ways now, and you look to see, what are the doses, if you will, of practice after a stroke that are necessary in order to both positively reorganize the brain and also positively shift behavior, the doses are fairly phenomenal. So, for example, this is the original Nudo study, and his monkeys did 9,600 movements, or actually retrievals of a banana-flavored pellet out of a very small little well, before they see, cortical reorganization. These are the data extracted from the Excite trial, which was a constraint-induced randomized control trial that finished up a couple years ago. And in this particular trial, patients used their hemiparetic arm 196 hours over a 2-week period. And 60 of these hours were in one-on-one therapy with OT or PT. So these are massive doses of practice. And we see the same thing if we just look at learning studies in healthy control and also in stroke patients. So you look at things like 2,400 repetitions of a 10-item sequence. This is 24,000 movements, 31,000 movements. Both of these are humans. Another animal study, which is 7,000. And in my own work, we really don't start to see changes in terms of fMRI data and or behavioral criteria for learning until we get to about 1,500 practice trials. And this is of a very simple motor sequence. So the issue then that we bump into here is how do we start to deliver these kinds of doses to our patients And are there ways to maybe get around delivering these massively high doses of practice to our patients that might not be so mind-numbing as doing something, 24,000 times? This is mind-numbing both for participant and therapist and or investigator alike. So we then have been trying to think about, are there ways that we can facilitate learning to get around this, what I would call dose problem, and to also then speed recovery of function. And at a very blunt level, if I kind of go through and start sorting out, our work, we come up with some lists of things that we know seem to be impediments to learning, and a converse list of things that we have found that seem to facilitate learning after stroke. So low dose of practice, and we've recently kind of qualified this or quantified this through a study that's coming out next month in Archives of Physical Medicine and Rehab. Instructions, the way that we set up practice sessions seems to have a very large effect on eventual kind of level of motor learning. Sequence or movement organization is also something that impedes recovery. And then altered force control seems to be a real impediment to how people with stroke can use their stroke-affected hand. There is a little bit of good news. We have a recent series of papers demonstrating that sleep has a very positive effect on motor learning after stroke. Task-specific training, so looking in a very, very specific fashion about how we learn or how we're practicing also seems to help. And then there's a question mark here. We think this is positive, but we're not sure of all the parameters or all the characteristics, so we're still quantifying, but I'll then move into brain stimulation and what we know and what we don't know there. So I want to just kind of walk through each of these and show you some of our data and hopefully convince you that we're making a little bit of headway here. So the first is, I think one of the main things that we can all agree on, at least in the therapeutic community, is that a low dose of practice is a huge problem. And it's one reason why our patients, when they come out of their acute or their inpatient rehabilitation phase, that they have not really maximized their potential. So we finished up last year a 9-site international observational trial that was headed by Catherine Lang at WashU in St. Louis. And basically what we did was very simple. We went out, and actually none of us went out. We had our research staff go out and sit in clinics and essentially count repetitions and say, what are we doing in rehab and what's the dose of what we're doing across different domains. And this is just one snippet of that. This is numbers of repetitions for the upper extremity, and we've categorized our practice by active exercise, passive exercise, things that involve sensory and things that involve functional movements. I'm sorry, my axis got a tiny bit shifted here when I brought this in. So essentially, as on a nutshell, you can just see very easily from this plot that the dose of practice in this session, at any rate, is incredibly low as compared to those excerpts that I just showed you. So for example, in most of my work, people get at least 500 practice trials in a day. And we're looking here now at someone with stroke who might be doing 50 repetitions of active exercise, and really most frighteningly about 25 repetitions of anything that's functional. And it's quite interesting, across the board, only about half of the sessions that looked at upper extremity function actually incorporated the use of functional movements into that rehabilitation session. So I think that kind of taken together, and it's a little bit hard to dispute, we would all agree that this dose issue is a huge one. And some of the real successes of, for example, these constraint-induced therapy trials come from the massive dose of practice that occurs in a relatively short period of time. So we've done also quite a bit of work over the years looking at what happens when you instruct someone how to move. And actually, this comes right out of a question I had years and years ago when I was a clinical physical therapist. And I was always very interested in understanding whether or not telling someone how to move actually translated into their movement. And so it turns out that after stroke, we use instructions very differently than healthy controls, but even healthy controls don't always benefit from instructions. And these are just 3 of the different studies we've done. We've looked at basal ganglia stroke, to a more global middle cerebral artery strokes, and then different kinds of task in these middle cerebral artery strokes to see if this is a task effect. But kind of summing up about 5 studies in one slide, essentially what we've found is that when you instruct someone about how to move, so you say, For example, in these studies, there's a repeating sequence. Here's what the sequence is. You can even let them memorize it in advance so they can explicitly or declaratively tell it to you, tell you the 3 steps of movement they're going to undertake. And you compare that to people who you just allow to learn. You just say, practice, do the best you can, be as fast and accurate as possible. We find a very interesting dissociation in what happens in healthy controls. And then these are the data from the basal ganglia stroke. So these are the explicitly instructed individuals, and these are the people without instructions. This is an RMS error score, so down is better. It means we're making less error on our tracking task. And what we essentially see is that healthy people in this particular task can benefit from instructions. The individuals with stroke perform much poorly when we instruct them. And we actually show that when we give instructions, they essentially stop improving. So we've done these studies where we take a day and we let them practice without instructions. And then we start giving them instructions about how to move, and we basically can totally impair their performance until we stop giving instructions, and then they start improving again. And these are just a retention test summary. So when we just allow these folks to just practice on their own, they perform much better. Actually, this is change, I'm sorry. So actually, up is better. That means they made more improvement from beginning to end. I'm at a funny angle on my slides, although I suppose I could look ahead. So I apologize for that. So kind of in a nutshell, we find that explicit instruction benefits healthy controls and interferes with stroke. And we've all had this experience of having instructions interfere with our motor performance. And so anyone who golfs in the audience knows this very well. So the worst thing anyone can ever do to you when you're golfing is give you advice right before you tee off, right? So, oh, what are you doing with your elbow? Maybe you should keep it a little straighter. Or try to, A, B, and C as you swing through. And if you ever are golfing and you want to be sure to win, give lots of instructions to your golfing partner as you kind of proceed through the course that day. And I promise you, you will screw them up infinitely. And this is a very classic psychological phenomenon. It's actually called the Bliss-Boder phenomenon. And so it's been shown many different times over the years, and we see it almost accentuated in the stroke population. And we actually think, kind of from a neurobiological perspective, when you have damage in especially the basal ganglia, what we have is an interference in the network that translates explicit information from the medial temporal lobe into motor action, which probably happens somewhere in the basal ganglia and or their associated circuitry. It's a little hard to know exactly where that happens given the incredible intricate loops that come in and out of the basal ganglia. But we do kind of consistently see that when that area of the brain is damaged, we have a lot of trouble with incorporating instruction into movement. Okay, so a more recent study that we just actually had accepted, I think it actually came out on Monday, was looking at when people with stroke do learn, and we do show learning in this population. So it's not that they can't learn, they just learn differently. We found that one of the problems that they're confronting is the inability to organize movements in the same fashion that you or I would. So there is a phenomenon in learning that we call chunking. And basically what this means is when you have a complex movement or a complex task you're encountering that has many different movements, as you learn the task, you group or you chunk individual movements into larger single motor plans. And the best example of this is learning to drive a stick shift car. So you're learning to drive a stick shift car, you have to push in the clutch, take your foot off the gas, you've got to move the stick shift, and then you've got to reverse that, right? You've got to put your foot on the gas and take your foot off the clutch. And when you very first learn that essentially is a 5-element movement. You program each of those movements individually. Then as you get better and better, you start to group, the gas and the clutch on the first, and then you shift and the gas and the clutch on the second. You might be down to three motor plans you're running. And eventually you're running that as one motor plan, which makes you faster and much more efficient. And so we actually looked to see if this might be part of the problem in people with stroke. And there's some other data to show that as we age, we're not as good at grouping our movements or chunking them. So this is a very complicated 3-dimensional serial response time task where people are learning to make a movement to a certain location, with a corresponding key press that goes with that location and also in a certain specified time. And these are stroke patients who are now using their non-hemiparetic arms. They're using their less affected stroke arm. We know that non-hemiparetic arm isn't normal, but it certainly is closer to normal than the stroke-affected arm. And so these are absolute reaction times, so down is better. This is practice session across time, and we have some random sequences and then repeated practice of a an implicitly learned motor sequence in there that the patients actually did gain awareness of, that they were learning a repeating sequence. And what's interesting is when you look at the random sequences here and here, you can see there's no real big difference between the healthy controls and the strokes. The healthy controls are faster, but this is not a statistically significant difference. Across time, though, you can see that the healthy controls become much faster. They make quite a bit more change than their age-matched counterparts with stroke. And when we do some hierarchical clustering analysis and we look at response times in early practice, so this would be block 2, you can see that this is in the black is a random sequence, and in the open circles here is the repeated sequence. There's no real difference between the two. So there's no real chunking happening here. Same for the stroke patients over here. They're really not different. When we look at the retention test, which is after 3 days of practice, here's our healthy controls. And so obviously, again, faster is better. And what we can find is that for the healthy controls, they are, through our cluster analysis, we identify clusters that are programmed together. So here would be one cluster, elements 1, 2, 3 of the sequence. There's a second cluster for 4 and 5. We have two individually programmed elements, and then a third cluster and a fourth cluster. So that healthy controls are running off the sequence with 1, 2, 3, 4, 5, 6 motor programs. You can contrast that then to the stroke-affected individuals who are now running the same sequence, which they have learned. They're significantly faster, but they're programming this with 1, 2, 3, 4, 5, 6, 7, 8, 9 motor programs. And so they're never as fast or as fluid and efficient in their motor production, despite the fact that they've learned. So this is maybe one other explanation for why we see these learning-related deficits in individuals who have had a stroke. And then kind of last on our bad list, and then we'll try to pick it up a bit and be more positive. We also have recently finished a series of studies looking at the control of force in the hemiparetic hand as compared to the non-hemiparetic hand. And these were actually studies, this study was done in the fMRI. And what we were looking at was how much percent signal change in different regions of interest of the brain, how strongly activated are those regions for controlling essentially the same level of force in the stroke hand as the non-stroke hand, and then in a series of age-matched controls. And so I have the contralateral regions of interest. So these would be the brain that's contralateral to the hemiparetic hand during a hemiparetic squeeze. And we have the healthy controls in the darker gray and the stroke in the lighter gray. And then we see essentially the same phenomenon ipsilaterally as well. But essentially what we see across the brain, both ipsilateral and contralateral, is to generate the same amount of force, we use, or the stroke-affected individual uses, quite a bit more neural resources, if you will. So they're working an awful lot harder in order to essentially generate the same force. So again, a very inefficient force production and force control strategy. And we actually consider that, given the higher levels of force across the movement, is they're almost saturating. So if we're at trying to squeeze at 30% force, they're using a very good chunk, for example, of the primary motor cortex. So they're just working an awful lot harder than are their non-affected counterparts. And what's interesting to me is that this isn't specific to the contralateral hemisphere. So we see essentially the same issue in the ipsilateral hemisphere. So for example, M1, you can see they're working significantly harder at M1, the individuals with stroke, than the healthy controls. So we may just kind of be running out of neural resources, and that may be one of the problems that they're confronting. Okay, so what things do we know seem to help learning and maybe recovery? And I meant to say early on that when I say learning or motor learning, I'm really thinking recovery, and I tend to use these two terms interchangeably because integral to any recovery process is essentially relearning lost skills and learning new ones. So one of the more exciting things that we've come across recently is some work done by my doctoral student, Catherine Siunkinsong, looking at the effect of sleep on implicit motor learning. And this is a really interesting story whereby individuals who are young and healthy, we know that when you learn a new factual or declarative skill, that sleep is absolutely essential. And there's a very robust literature suggesting that. So all of you residents do try to sleep on occasion if you can. Otherwise, it's kind of for naught because you're not really encoding that information. As we age, the story becomes a little murkier in terms of sleep and declarative learning. In terms of implicit or motor learning, sleep in at least young healthy people doesn't seem to have a large effect. So it seems to be that in order to actually learn a skill, you need to practice it and allow a period of time to pass for that skill to be consolidated into longer term memory. And then when you come back, whether or not you slept in that intervening time, you can still show a demonstration of preserved skill acquisition. So we tested to see whether or not that was also true in people with stroke. And we essentially had people practice a motor skill, and they then either waited 12 hours and had a retention test or had their normal overnight sleep. And then we gave a retention test the next morning. And we actually were quite startled at our findings. So this is an RMS error score. And what we're looking at is change overnight or between the practice and the retention test. So this is what we call offline motor learning. So no practice has happened in this intervening time. This is the difference between the last block of practice and a follow-up retention test the next day. We have 4 groups. We have a stroke group who slept, a stroke group who didn't sleep, age-matched healthy controls who slept, and age-matched healthy controls who didn't sleep. And what we found was this very large effect of offline learning in the stroke group who slept. So this essentially means that we're continuing to gain skill overnight in our sleep, such that in the morning we perform better than we did before we went, at the conclusion of our practice session, which happened before we went to sleep the night before. If you just wait the equivalent amount of time, you see just a little bit of offline learning, but not much. And then essentially, there's no real difference between waiting or sleeping for the age-matched healthy controls. So this, suggests that essentially that in the stroke-affected brain, sleep is performing some kind of an essential function, helping to encode learning and encode new skills. And we now are following this up, obviously, with some sleep lab studies. And kind of at an early stage, what we're actually seeing is that the sleep patterns of individuals with stroke resemble more closely the sleep patterns of young healthy people than they resemble the age-matched controls. So unfortunately for all of us, as we age, we spend less time in REM sleep, and we have fewer cycles between REM and non-REM through the night. And that is maybe one of the reasons why learning, for example, drops off a little bit as we age, at least motor learning does. The stroke individuals in this study, at any rate, are sleeping, and their sleep is looking quite a bit more like that of a young person in that they have more, statistically, a more percent time in REM sleep. They have more cycles between REM and non-REM. And it may be that it's this cycling between the phase of REM and non-REM sleep that's actually pretty important for motor learning. And so this is now being followed up on a much larger scale in conjunction with the sleep lab. So we'll see if those findings bear out or not. But it is very interesting, and it's very clinically relevant in that one of the easiest things we might be able to do for our patients is help them sleep and allow them to sleep when they actually are in the hospital, which, as all of us know, is a terrible place to get any sleep if you're actually the patient. We recently also have been looking at constructing practice. And one of the things that also seems to be emerging is that the training that our stroke patients engage in needs to be very specific to the tasks that they're trying to perform. And so in this study, we actually were looking closely at a phenomenon that's seen commonly after stroke, and that is of bihemispheric motor cortical activation. And so what you see in many stroke patients, somewhere between 75 and 90% of people with middle cerebral artery lesions, is that even when they're not showing any mirror movements as evidenced by silent EMG essentially, we see that both motor cortical areas are active when the hemiparetic hand is moved. So we took individuals who we knew demonstrated this pattern, and we randomly divided them into 2 groups. One group practiced an experimental task very specifically, and the other group increased their arm use, but it was in a generalized fashion. So they did things that weren't specific to the task being learned. And the task they were learning is this discrete serial movement task. It looks a lot like your robot. That's something I need to talk to you about. It's almost the same task. And we did a regions of interest analysis, just essentially trying to understand, does task-specific training essentially shift the pattern of brain activation more towards normal? That is, are we using more just contralateral motor cortex, for example, as opposed to a bilateral? And we capture this through just an index of excitability across the cortexes called the laterality index. So what we're looking for is a laterality index that's moving towards 1, which means we're shifting closer to using that contralateral cortex. So these are just some individual subject examples. This is someone who trained in the task-specific group, and here's someone who trained with just increasing generalized arm use before and after training. These, of course, are my star pupils, so take it with a grain of salt. But what we see, though, is that, for example, if you look at this post session here, what we see is a shift away from using the ipsilateral cortex. This is pretty much the same slice, so as evidenced by less activation here, and towards contralateral cortex. And you really don't see that shift in the terms of the pattern of activation across the generalized practice group. And we then can quantify those data with our laterality index and show that for the task-specific training group, we essentially were equally activating the hemispheres before training and following training. For the repeated sequence, which is the sequence that they've learned, we've shifted towards a more contralateral activation. That is not true for random or non-learned elements of the sequence. So when you practice random elements of movement, you may be improving your motor control, your motor execution, but you haven't learned anything. And so there's a distinct difference depending upon whether or not the sequence has been learned. And that's also then true for the general practice group, who essentially don't really learn our motor task because they haven't practiced it. And despite the fact they've used their arm a lot more, we don't see a shift in this activation pattern. And then kind of as a transition then, the last thing that we think may help learning is brain stimulation. And by this, I mean transcranial magnetic stimulation. So kind of, again, coming back to this dose problem, part of what we've been thinking about is there a way that we might prime or prepare the brain to learn such that we're now working with a more optimized system? And that's essentially what TMS allows us to do. And so just kind of as a really brief overview in case you're not all very familiar with this. So TMS is a method where we can externally induce and using an electromagnetic induction technique, excitability into the cortex. And what we actually are doing here is exciting the horizontal fibers in the cortex, which then subsequently affect, if you're in M1, the pyramidal or the cortical spinal tract neurons. What you get then on the output side, or ways that we can kind of index that we've actually done something with this electromagnetic current, is we can see changes in EEG. Many studies have shown changes in blood flow metabolism by PET or fMRI. We, in the lab, visibly see and record muscle twitch changes, EMG. And then, of course, our focus is in looking at the behavioral changes that are associated with TMS. This is kind of a nice study that actually we're trying to replicate right now, whereby you can essentially concurrently, and there's not much that you can record concurrently with TMS because it's such a relatively large electromagnetic current. It usually blows out channels, for example, of EMG, and it's ferrous, so it's hard to get them in the fMRI, although not impossible. But this is a near-infrared system. And you actually, if you are applying some pulses of TMS and pairing that with behavior, and then measuring by near-infrared, you do see these changes in hemodynamic response that are fairly immediate. So this is oxygenated hemoglobin in the red. And at time 0, there's paired finger tapping and TMS. And in this particular illustration of this by Hada et al., what you see is an immediate increase in oxygenated hemoglobin directly under the coil itself. So this may be one of the ways that TMS has its effect. It's a little bit unknown exactly what all of the different effects are of TMS. And there's quite a bit of work underway to try to understand this. So we actually right now are replicating this study. You have to invite me back in 2 years. and I'll tell you all about it. It's a little bit of a tease, I guess. We have now, this is a 2-channel NIRS system. We have a 32-channel. So we're trying to look not just under the coil, but at more whole brain changes that might be induced by exciting one element of a motor network, and then looking to see how that changes across time. But we do know that we're doing something to the hemodynamics directly under the coil that's fairly concurrent in time of stimulation. The question is, this is a single pulse study, so they've just given, one zap and then had some behavior. And the real question that I have here, I have two actually. One is, when we do a repetitive TMS study and we do a 15-minute bout of TMS, what's the duration of that after effect in the terms of the changes of the hemodynamics? I don't think that's entirely clear yet at this point in time. And another question that I, of course, have is, well, this is in a healthy brain, so what happens in someone who has really screwed up hemodynamics, which would be our stroke patients? And do we see a similar pattern, or is it different? So globally or generally, there's two kinds of TMS. We have pulsed or single and paired pulse TMS. This is the type of TMS that we use to do things like establish motor thresholds. And a motor threshold is essentially just the lowest TMS intensity that will reliably induce a change or a twitch that we can see by EMG. And this is called a motor evoked potential. And one of the reasons why this is kind of important is everything in TMS is essentially normalized to each individual's motor evoked potential. Our motor evoked potentials tend to be fairly stable across time. Mine's about 48 on any given day. If I'm really caffeinated, I might be able to drop it down a little bit. And that's 48% of stimulator output. So it's not like 48 volts, or I can't give you a unit on that. But we do normalize everything to this threshold value for essentially eliciting a muscle response in the muscle of interest that we're working on. Pulse TMS is useful for mapping. Motor evoked changes in MEPs can be shown before and after treatments, before and after drugs. This is the kind of information that you collect when we're doing things like looking at central conduction time. So how long does it take for between the time we put in a pulse till we see this response in the EMG in the periphery. And that can be a very useful tool, for example, in looking at patients with multiple sclerosis. So we're doing a study right now with our MS clinic looking at are people progressing or regressing based on their central conduction time. More kind of fun is the repetitive TMS. And so we can do two things that are critically determined by frequency with our TMS. So this is when we run a train of pulses into the brain. So if we run these at a frequency above 5 hertz, we can excite cortex. And if we run them at a frequency below 1 hertz, we can essentially inhibit or virtually lesion the cortex and have somewhere between a 15 to 30 minute after effect window by which we can kind of look at different cortical changes, either in excitatory or inhibitory circuits. The repetitive TMS is an offline effect, and it's critically determined both by this frequency and then the intensity which we run it at. But we almost typically, apply the TMS first and then follow it by some kind of behavior. And we think this duration lasts between 20 minutes and an hour. There's another kind of repetitive TMS that we're now working with quite a bit, and it's called theta burst. This is essentially a way of running our TMS at an incredibly high rate, about 50 hertz, and giving kind of rapid bursts of this high frequency stimulation. The higher the frequency of the stimulation, it almost seems like we get a better, a stronger, and a more robust effect. The problem is we're also much more likely to cause a seizure at high frequency. And so theta burst is kind of a way around that. So we give a very quick, for example, 5 millisecond burst of 50 hertz stimulation followed by a 200 millisecond window. And then we burst it again. So theta burst is interesting in that we seem to get a more reliable and robust effect, especially when we're trying to inhibit systems. And that's been shown quite a bit in other people's literature. And I have some data I'll show you in a second that suggests the same. The other nice thing about theta burst for inpatient people such as myself is one way that we index the dose of TMS is how many pulses we put into the brain. So in order to essentially get 1,200 pulses of 1 hertz TMS into the brain, it takes us about 20 minutes. We can get the same dosage in 40 seconds if we run continuous theta burst stimulation. So it's a little bit of a time thing too. And this is friendlier than on the patient and on the applicator. So you can use TMS in lots of different ways. It's been used to try to predict recovery after stroke. We can map experiments. We can look at relationships between the hemispheres, which we're doing quite a bit of, and it's a very interesting story. You can obviously give transient or virtual lesions. We can transiently excite. And then we in my lab are trying to pair excitation and inhibition effects with motor learning. And what TMS, I want to emphasize, is not a silver bullet. We don't just give someone a TMS and then send them out and say good luck. There's quite a bit of literature that has tried that, and you don't nearly get the same effect as if you apply your TMS and then, lo and behold, pair it with some kind of behavior. This maximizes your effect. And so it seems to be that it's kind of the 1-2 punch that seems to facilitate learning. Of course, there's a very huge question in, for example, stroke recovery is, well, where do we stimulate? What's the area that's of most interest in terms of recovery? And then very quickly, I mentioned already, but the one thing that we worry about the most in my lab is someone having a seizure. This actually is a fairly low risk, except for people who have any kind of history of epilepsy and are on medications. And there are now, there's been several consensus papers that have suggested safety standards to the body of researchers looking at TMS. And when those safety standards are following it, the risk of seizure is significantly lowered. And in fact, there have been no reports of seizures in the last now 10 years since the safety standards were implemented in people who were following the safety standards. So there seems to be some kind of critical interactions between frequency and dose and intensity that we can manipulate to keep our patients safe. We still worry about it, though. The only other effects of TMS that we seem to find is some people will get a headache. It's not very common, but it does happen, and it's a little uncomfortable under the coil, more uncomfortable for some people than others. And it's actually quite loud, so we take quite a bit of care to protect our hearing and our patients' hearing. So some of the things we know about TMS with respect to stroke that are very exciting is that there's a lovely paper in Brain by Kathy Stenier about 1.5 years ago now. And her group essentially demonstrated that the presence of a motor evoked potential, so the ability to elicit a twitch with TMS, essentially tells you whether or not the cortical spinal tract has its integrity maintained after stroke, and is one of the best predictors of whether or not that patient is going to respond to any kind of rehabilitation treatment. And in this same paper, she essentially showed that, with a 5-minute TMS mapping session, you can get essentially the same data as you would from an anatomic MRI, a functional MRI, and diffusion tensor imaging. So here's 2 patients to contrast really quickly. This would be the affected cortex. This is the unaffected cortex. And she's doing something called paired pulse mapping here, where you apply a conditioning burst and then you look to see at what the resultant response of the brain is to a subsequent burst. So you can see in this patient, when you stimulate them, you get a response from both stimuli on both the affected and unaffected hemisphere. Here's another patient, contrast that, where you see no response to the stimulation, so cortical spinal tract is not intact here. And then on the unaffected side, a normal response. Here would be their lesions, so a lesion in the internal capsule on the patient with no response, on the patient with a response, but not a full lesion. It's interesting and classic. Here's a much even smaller lesion, but poorly located, if you will, and blocking the descending tracts as they come through the subcortex here in the patient that doesn't have the MEP response. The patient with a normal MEP shows a fairly normal pattern of brain activation during a hand squeeze task. And here's their diffusion tensor imaging data showing that the cortical spinal tract has its integrity. On the converse, again, here's the same patient that showed that they did not have the motor evoked potential. They show an abnormal pattern of cortical activity. And you can see clearly here that there's a significant loss of the descending tracts as shown by DTI. So you can get essentially the same information or complementary information from your TMS measure as you would from all these other kind of high-end, expensive, time and labor-intensive measures. So some of the stuff that we're working on to try to think about motor learning and brain stimulation, it involves kind of trying to understand which side of the brain to stimulate. So do we get more of an impact if we stimulate contralaterally as opposed to ipsilaterally to the hand being used? And then also looking at some different areas. We kind of have a combination of things that we're investigating. So the first, and this is a study that's actually out for review right now. So we asked participants, and these are now healthy participants, and then I'll show you how we're using this in stroke. These are participants that we asked to learn our continuous tracking task. And essentially in this particular task, you are tracking a target as it moves up and down on the screen. The target's the open circle, and your job is to put the red dot in the open circle. It's an impossible task. No matter how close you get, this thing squirts away. Patients hate me, and so do participants. So you practice periods where this is moving randomly. Embedded within each random, there's always a repeated sequence you practice over time, and then you move randomly again. And what we look at is root mean square error, and essentially it's a very robust task. What we see is if we compare error on the random to error on the repeated, we find that people become much more accurate at tracking the repeated sequence despite the fact they're never aware that it's there. Now, we targeted the dorsal premotor cortex here. Dorsal premotor is a very interesting area for lots of reasons. We think it's very important in motor planning. So classic animal work shows that when an animal is told that it's going to make a movement but given a delay cue, dorsal premotor is very active during the delay. And we think it's holding and forming the motor plan. And then once the animal's allowed to actually make the movement, it becomes silent and activity shifts more towards motor cortex. The other reason we're interested in dorsal premotor is it's been shown in imaging and animal work to be an area that has a lot of substrates that we think may be important for the recovery of motor function after stroke. There are cortical spinal tract neurons with direct connections to the spinal cord in dorsal premotor. And there's some really nice work, again, out of Randy Nuno's lab showing that when you lesion M1, that the magnitude of the lesion in M1 in terms of percent territory lost is inversely related to an expansion in premotor, such that the more M1 lost, the more expansion in the premotor cortex you see. And we see lots of activity in this area of the brain in fMRI in patients who are recovering and recovered. So we have a stereotaxic system in my lab called BrainSight that allows us to co-register the application of our TMS, and this is the TMS coil, actually with the anatomical MRI of the participant. And this is really nice because it allows us to precisely target, yeah, this is actually my brain. It allows us to precisely target different areas of the brain that are outside of motor cortex and to be very precise in terms of repeated sessions of TMS. Not just in terms of the location, so this is my dorsal premotor, but also in terms of the orientation of our coil in three-dimensional space. So you can see it'd be quite easy. This is a stimulation stick, essentially, representing the direction of the stimulation. And it's really easy when you're holding this coil externally to drift in any of three-dimensional space and actually pretty significantly alter where you're stimulating. So it's pretty easy when you're on dorsal premotor to kind of fall back and all of a sudden be hitting M1. So this is a really invaluable tool for us. So we targeted, again, the premotor cortex and had some kind of interesting findings here. And these are our healthy control people. And we did this kind of as a proof of concept before we repeated this experiment in people with stroke. But essentially what we found is that we had three groups: excitatory repetitive TMS, inhibitory TMS, and a sham TMS. We have a TMS coil that looks and sounds identical to our regular coil. We've actually then added onto it a little electrical stimulation electrode so that you get the same kind of scalp tingly thing, but there's no current coming into the brain. Our patients and our participants are kind of sophisticated, so we have to fool them as much as possible. What we find is quite interesting. So in this study, people got TMS and then they immediately practiced. And if you look at both repeated and random sequences, and again, this is root mean square error, so down is better. We had 4 days of practice and then a retention test, which I'm going to talk about in just a second. But across practice, at any rate, there's no difference between the groups that get excitatory, inhibitory, or sham TMS. And that's true either for repeated or for random. So on the repeated sequence, everyone's getting better. Interestingly, when people come back to see us for a delayed retention test, and our retention test day, we don't do any TMS. We just look to see, what happened and what's their skill in the absence of whatever manipulation we've been working with. We find that all three of our groups, if you compare RMS error on random with repeated sequence, we see less error on the random. This is a classic hallmark that they've learned, the sequence they practiced. We see even less error in the excitatory group, though, than we do in the inhibitory, the sham. So the group that we've excited their dorsal premotor, they're even more accurate than the other two. And again, what's even most striking is that a significant amount of this improvement actually occurs offline. So this is just like the sleep data I showed you before, whereby between the last block of practice and the retention test the following day, we see that the group in the excitatory TMS is improving overnight. And so this is one of the things that in motor learning we say suggests something going on with motor consolidation. So in some way the motor plan is being updated and becoming more stable overnight. Everyone slept, so we don't think this is a sleep-related effect. But we do show this very unique response, and this is related somehow to having pre-excited or primed dorsal premotor cortex. So it's pretty interesting. It, of course, is not at all what we expected to find, because it is research. And so that's just kind of seems to be the way it goes most of the time. We really thought we'd see a difference in practice, but it seems to be when we kind of step away and allow the brain to consolidate the skill. We're now rerunning this study actually again in healthy controls, because what we did every day was the person had their TMS and then practiced, came back the next day, had their TMS and practiced. And so we may have been having this offline learning effect every night, but we kind of blew it out of the water with our stim. So we're now redoing the study where they get their stimulation, they practice on day 1. They have a retention test at the beginning of day 2, get their stimulation practice, and so we go from there. Okay, so we now have 3 other things we're working on. One is what happens if we inhibit ipsilateral primary sensory cortex. There's some very interesting interhemispheric interactions after stroke. Such that the stroke-affected hemisphere seems to be very dominated by inhibitory circuitry and neurotransmitters, and that thereby essentially releases the inhibition that is normally transferred transcallosally to the non-hemiparetic cortex, and this cortex then becomes overactive. And so one idea that we have had is, well, could you come in and essentially inhibit, essentially over here, this contralesional, could you inhibit the cortex ipsilateral to the hand being used after stroke and show an effect on the overall performance of the individual? So we essentially have looked at this very carefully, and using EEG targeted the transcallosal effect of inhibiting, this would be left sensory cortex, and then looked at right hand function. And this is just our actually EMG signal from the P4 electrode. And let me pop to one that's easier to show you. So we compared 1 Hertz repetitive TMS to our theta burst. And what we essentially show is that if we inhibit the left sensory cortex, we show larger sensory evoked potentials on the right sensory cortex. So even in healthy people, we can induce this transcallosal effect. So, and you guessed it, we're now, of course, doing this in our stroke patients, and then pairing that with behavioral motor learning. Another thing that we've done that I'm very excited about with our TMS is we've been able to confirm some experimental findings in stroke. And so briefly, one of my other doctoral students, Eric Vidoni, did a very nice series of studies whereby he looked at the need for proprioception in motor learning. So first off, he took healthy control people and he vibrated their arm, which is essentially interferes with your proprioception. He then captured these interferences by doing a limb position matching task and showing these decrements in proprioceptive accuracy when the arm is vibrated. He then looked at whether or not this affects motor learning. And so when you vibrate young healthy people and you disrupt their sensation peripherally, what you see is that there's changes across practice, but at a retention test, when the vibration is removed, there's no difference between a group that was vibrated and a group that was not. He then repeated this experiment in stroke patients, but we don't need to vibrate them because we already know their proprioception is disrupted. And we essentially show a relationship between limb position matching accuracy and change across learning, such that people with better limb position matching accuracy, which suggests proprioceptive integrity, show more change when they learn. So you need proprioception for learning, but it seems to be that central proprioception is what's important, so the central processing of proprioception. So we just have now rerun this experiment in healthy people where we can actually virtually lesion S1 and see what happens. So when we lesion these people, we show decrements in limb, when we give repetitive TMS to give a lesion to healthy controls, we see decrements in proprioception. We see increases in their 2-point discrimination. Very interestingly, if you look at their data across practice, they look just like our they look just like our stroke patients did, where they show improvements across practice, but in a non-specific fashion, so repeated and random, aren't significantly different. And when we compare those who were stimulated to those in a sham group, and we look at just improvement or change in root mean square error, you can see that the group who got sham stimulation is doing quite a bit better. So we showed more change in their motor behavior as compared to the experimental group. So this is a really nice verification of our stroke lesion model, and how it's really the central aspects of proprioception that seem to be important. So then one last thing that we are looking at, and that is whether or not we can actually try to understand who's going to subsequently have a stroke based on their interhemispheric excitability after a TIA, or a transient ischemic attack. So there's a method by which we can essentially index the circuitry of the brain and the relationship in terms of intracortical excitability, and essentially the ratio of excitability to inhibitory circuitry. I would be happy to explain this technique afterwards. It's a little bit complicated. But essentially, this is the way that we're able to capture this interhemispheric disruption, such that we see that the stroke-affected hemisphere is much more dominated by inhibitory circuits, which actually we think are related to GABA. And this is a paired pulse recruitment curve, essentially showing the inhibitory side of the curve and then the excitatory side of the curve. And some nice drug studies done by some other investigators have shown that this inhibitory side of the curve is related to GABA, and the excitatory part of the curve is related to glutamate. So in stroke patients, we see that this curve is shifted such that you see much more upregulation in the stroke-affected hemisphere of the GABA circuits. So we now are essentially doing the same thing in patients with TIA, and it's very interesting. There's one other report showing that even 7 days after a transient ischemic attack, which, as we all know, is something that resolves very quickly in terms of our clinical symptomology, but even 7 days out, we still find that the brain of an individual with TIA is showing these intracortical asymmetries in the same fashion as someone with stroke would. So obviously the brain is not normal, even though clinically their symptoms are no longer manifesting themselves. This is now an asymmetry index between the two hemispheres, showing that the TIA-affected hemisphere is much more dominated by inhibition. And there are actually some changes in facilitation as well. So I'm working with some very patient epidemiologists on this study. So we essentially collect this data, and now we're just waiting to see if any of these people stroke. It actually is kind of killing me, because I'm not used to having to wait. And I also feel kind of terrible about running around hoping someone's going to have a stroke. But the question is whether or not the magnitude of this offset in intracortical asymmetry will predict both the incidence and the subsequent severity of stroke in the outset. And it's really interesting, because we're doing a follow-up study now to see how long this asymmetry persists. And in some patients, we can see it 14 to 21 days after TIA. So it's a very longer term effect on the brain than we actually can index clinically. So it's pretty interesting, and we'll see what happens. So just to kind of wrap up then, I think that TMS is a useful adjunct tool for indexing changes in brain state and stimulating some changes and then looking at those effects. We have a lot of questions left. So is it a good predictive tool, for example, after TIA? And can we use it to promote recovery from stroke? We have 3 stroke trials underway. We're looking at stimulating premotor on the ipsilesional side, and then we're doing 2 contralesional studies, one looking at M1, one looking at S1. And I'm not sure of the answer here. The answer about where we stimulate and how we stimulate, maybe it depends. And I think it may depend a lot upon the patient themselves and their lesion location and their severity. And so there's a lot of unknowns here. But it seems promising, so we'll see. So really quickly, I really need to thank my students, who really are the ones who do most of this work. And hopefully they're all, well, it's a little early, but shortly here, I'm sure they'll be slaving away in my lab. And then, of course, the people who pay for it should be acknowledged. And then last but not least, I should invite you all to beautiful British Columbia. We are hosting the Winter Olympics next summer. I don't actually do this, but it looks really cool. And yes, this is ice. You guys could do that here, right? I'm sure some of you do out on the lake. No.
Theory is far too dangerous.
Okay, so maybe not. This is a smaller lake, but It's a lovely place, and if any of you are ever in the neighborhood, you should certainly stop by the university. This is actually the view from the university, so when I visited, it didn't take a whole lot to recruit me there. I was like, okay, happy to. So I'd be happy to entertain your questions if you have time to stay.
So on behalf of the two Centers of
you.
Excellence, a little token
Very
of our
nice. Thank
appreciation.
you.
And let's hear questions. Excuse me.
Yeah.
So I have one question, which actually I have a whole bunch, but I'll just ask one.
I have a whole bunch too.
I thought it was interesting that an early slide you presented suggested that for some stroke patients that instruction may be counterproductive. Mm-hmm. And I wonder if part of that relates to the instruction being something that would work, a scheme that would work for an able-bodied person, but in the stroke person there may be a block in carrying out that scheme. And so it's sort of trying to get the nervous system to do something that physically can't.
Yeah, I think that you may well be onto something. So the instruction story is incredibly complicated. And there's just a tremendous amount of work to be done in terms of thinking about the content of instructions, the timing of instructions. Are there ways to give instructions that aren't verbal? Can you model, for example? And then if you do model, is it best to model a perfect performance or an abnormal performance? So the instructions that we gave were things that those patients should have physically been able to do. And I'm realizing now one piece that I forgot to tell you about that was these were stroke patients using their non-hemiparetic arm. So in theory, if I say press, the red key, the blue key, the green key, there should not be a problem with that. However, there is, apparently, this translation is lost. So it could well be that we're instructing them to do something that they physically can't do. What I think is more likely, but this is my own theory, and it's a little hard to test experimentally, is the fact that what you see is this inability to transfer. And there's been some really nice fMRI data in very rapid time by Russ Poldrak at UCLA, looking at what happens when you're giving instructions about motor versus cognitive or factual types of things. And what you see is this kind of rapid alternation between hippocampal regions and basal ganglia regions. And the area that comes on in between is dorsolateral prefrontal cortex, is maybe a mediator kind of round the horn, if you will. And neuroanatomically, that makes a lot of sense. So I wonder a little bit if it's just a disruption in this basal ganglia that I can't take that information in.
Where I was actually thinking not so much they couldn't physically do it, but that the area that you're in, that you're calling upon, may have been recruited for another purpose.
Yeah, well, that could be too. It's interesting though, when you don't instruct them, they can do it. And so something I've wanted to do for years, and I need to get around to it, is to essentially redo this study over time in the fMRI and see, are they indeed bringing in this area in the same fashion or not? It would be a really neat study. So if one of you has the time and the resources, let me know. Yeah.
I wonder if you can comment on the failure of North Star when they try to stimulate the cortex for stroke patients.
Yeah, I have a lot of opinions about North Star. So the North Star trial, for those of you who may not be familiar, was an attempt to essentially sew into the dura a low stimulating grid. And they located that grid over the hand motor area and basically turned it on during rehab and then looked to see what the outcomes were. So I think you have a couple things going on there. There were no controls in that study for intensity of stimulation, for one. Everyone just got the same. And we know very well from the TMS work that our kind of thresholds are very, very different from person to person. They're wildly different. If you stimulate everyone in my lab, I have one student who I swear we can look at her and she twitches. I mean, her threshold is so low and we go all the way up on the other end. So I think that that's one real problem. Their stimulation parameters were not well optimized. I also think that M1 is it's the lowest hanging fruit. It's the easiest place to stimulate, but it's certainly not the most promising. So, for example, our thinking in looking at sensory cortex is if I stimulate sensory cortex ipsilaterally, I get an effect on M1 because they're reciprocally interconnected. I get an effect on contralateral M1, and I also get an effect on contralateral S1. So now I've brought in a big network as opposed to just M1. So I think that some of those things hampered them as well. And then last but not least, those people went through a huge neurosurgery to put it in and take it out. I don't think you can minimize those effects either. So yeah, I think a lot went on in that trial. Yeah.
It's very innovative work, and certainly reflects the challenges we see with stroke. You know, and with acute stroke, we talk about a therapeutic window, which is very short, maybe 3 to 6 hours. Do you have a therapeutic window for restorative therapies? And my second question is this modality specific? So, as you know, I mean, some have used a transcranial laser, for example, to stimulate the cortex. I mean, is this a non-specific stimulatory phenomenon, or is it specific to the modality?
Two really good questions. I don't think we have enough evidence to answer the first. I just don't. I think the field is far too juvenile. And there's many different ways. There's TDCS, I didn't talk about that at all, where you put a direct current in. And I don't think we understand the true network effects. So we're starting to do some work to try to pair TMS and fMRI to just understand if we hit one area, what's the broader approach. And the last failing, and I'm still talking about your second one, is we don't have a great animal model to really precisely categorize our effects for the laser versus TDCS versus TMS.
Do you see anatomic changes, new synaptic connections, et cetera?
Don't know, again, because most of this work is in humans. So there is a cat model they're working on. They have trouble with rodents and TMS. They can't get the coil small enough without melting it, the coil itself, not the animal, thankfully. So there's some work along those lines, but it's not out yet. And they've been working on it for quite some time, so I think they must be having trouble, is always my take on that. In terms of a therapeutic window, you know, I think it's the same as we operationalize in all of our work. So for example, in Dr. Daly's and everyone else's, you know, earlier is better. No one is doing any TMS work in anyone with stroke who's less than 6 months out because we're terrified of inducing a seizure. And so I couldn't tell you anything about that period of work back, or that period of time back, in terms of it being a window. And I don't think we're going to be able to do that until we really understand a little bit better the physiologic effects that we're causing and the optimized kind of parameters that we want to use for those.
So all the patients have stroke more than 6 months?
More than 6 months. We're looking, and I should have said that up front, it's chronic stroke.
The exact opposite of what we would do acutely, where the shorter the window, the better.
I know. And we just haven't done that work yet. I mean, the work, a little bit, it depends on where you are and how aggressive you can be with those therapies. Actually, TMS work is quite difficult in the United States. Based on my 2 experiences, you get to deal with the FDA on a regular basis, which isn't very much fun, and it's very slow. In Canada, I'm not Canadian yet, but I'm working on it, and it's not a very litigious society, so Health Canada doesn't really care that we're blasting away into people's brains. Now, we certainly ethically don't want to cause any harm, but I don't have to get every single protocol approved by some federal body before I can work. So I think that has slowed things down in a nutshell in terms of what people have been able to do here in the States in terms of pushing this window back. But now we do see effects in the chronic patient, and the effects are positive. So I think there's no reason to expect we wouldn't show something similar. And we would have less, if you will, learned non-use and negative cortical reorganization to fight our way out of. So in that sense, it would be probably easily.
Let others ask, but I mean, clinically, we know if we don't start just routine rehab while the patient's still in the hospital, they don't do as well.
Exactly.
And I suspect it's going to be the same.
I suspect it'll be the same too, yeah. Yes?
Generally speaking, in motor learning, there is data to show that treating animals pre-lesioning with corticosteroids actually shortens the duration of recovery and also the end level of recovery. And I'm just wondering if how would that integrate with what you're saying?
That's a very interesting question, and I'm not sure that I know. I suspect we would see similar effects, but I don't know of an experimental model that's explicitly tested that question with respect to either motor learning and or the TMS work. So I know that, for example, in Nudo's lab, they've done quite a bit, not with corticosteroids, but with amphetamines, and shown some really interesting results in terms of learning, 1, and recovery, 2, in the squirrel monkey model, based on the use of amphetamines, not before, but paired directly with the motor experience. So I'm not quite sure. That work has been much more carefully approached in humans. Although I suppose there's probably some case studies out there that weren't experimental that we could maybe look at in terms of amphetamine use and skill acquisition and learning. But it's not something that we've done any work with. We haven't done any drug trials per se at all in my lab. We're moving into some with the MS population, but it's quite a different setting.
The work is done predominantly in vestibular lesioning and motor learning, post-unilateral deficits.
Yeah, it's quite
Actually,
interesting.
Clinically, use of corticosteroids acutely can improve the recovery function and shorten the duration.
I don't know of a corticosteroid trial in stroke. Do you, Janis? I don't think it's been completely underexplored. I can't think of one. Yeah. Someone back there I saw raise their hand. Yeah.
Question. Would pantomime work better than explicit instruction?
Oh, yeah. It's a great and very interesting idea. So the question is, would pantomime work better? And there's some suggestion from the healthy literature and from one trial in stroke that actually observation of someone performing is a very excellent way to instruct about movement. There's a caveat. So when you do skill role modeling, watching an expert isn't helpful. So like it's cool to watch Tiger Woods, right? But it's really not helping me. I mean, I still suck at golf. It's not gonna assist me. But watching someone who's at a similar skill level make and then correct an error seems to be the most beneficial kind of pantomime or role modeling experience for motor skill acquisition. And that is in some of our work something that we've suggested as an alternative approach is kind of at a clinical level. You take a physical therapist and you say, okay, you can't give instructions, you need to be quiet. First of all, they can't stand it. It makes them crazy because they're used to talking all day. And then secondly, they're not sure what to do, and this is an excellent kind of adjunctive tool that might be useful there. So, yeah. Yes?
I was wondering if you could comment on what you believe is primarily affected by stroke. Is it the inability to form an accurate internal model of the task and subsequently a motor plan, or is it the inability to execute the motor plan because you can't produce accurate forces?
It's the question we wrestle with. So can they not execute the motor plan or can they not form the motor plan? I actually think it tends to be more towards you can't form a motor plan because we see so many deficits in the presumably non-affected side. I don't know that you can say it's all one or the other, but if I had to weight it, I would weight towards the one. And it's pretty interesting because some of the things that Dr. Daly's doing with having people move with the robot assisting them, it's aiding the sensory experience, which then is, helping to, I think, form a more robust motor plan, or some of our sensory stimulation that upregulates the sensory cortex also seems to facilitate skill acquisition. And so I have this feeling that it's something towards either forming an incomplete motor plan or forming it based on very strange sensory experience that is one of the large elements of the problem. Execution certainly plays into that, but even when we try to remove execution, so we practice random and repeated sequences, the execution elements are the same. For one versus the other, but we still are not normal, if you will, even on the stroke-affected side when we, normalize for motor execution deficit. So we subtract the random performance from the repeated. There still is a deficit there. So.
I just have to comment.
Yeah.
We're using this word stroke like it's one condition.
Thank you.
It's the heterogeneity of stroke is one of the big problems of the field.
It is.
The answer to that question is going to depend on where the lesion is. A lacume in the base of the pons is quite different than wiping out your middle cerebral arteries.
It's true. It's what keeps it entertaining, I suppose, for those of us in the field. And in a lot of these studies, we work really, really hard to try to have some degree of homogeneity, which means in stroke parlance, everyone had their lesion in the subcortex. Woo, that's a very homogeneous group, even though some it's putamen, some it's caudate, some it's internal capsule, some it's all of the above. But as a general response, I think it's more the formation. I don't know. Others may disagree.
Well, I was just trying to figure out the role of force production. Mm-hmm. We spoke briefly about it, but since the muscular talk was focused on coordination, at least I assume that the ability to produce forces is a major play.
Yeah, and it's interesting in that force production study, again, even the non-affected stroke hand is pretty messed up in its ability to produce and modulate force. And that is actually the first study that we've done in my lab, kind of foraying into the force realm. It's a very different area, and my collaborator on that study, Janis, is an expert in force production, and she drugged me there, kind of willingly, kind of not. But it is different, and again, though, any kind of modulations they have to squeeze and hold, again, that's dependent upon some representation and adaptation based on, you know, peripheral sensory feedback, and it's actually a fairly elegant act, taking all of that into consideration. But again, it's one of those questions, too, that I'm not sure we'll ever actually know the answer to, and it may depend in large part upon lesion location, chronicity, some other factors. Yeah. If anyone's in the middle trying to ask me a question, I apologize, because I'm actually totally blinded, so you'd have to yell and jump up and down. Yes?
So this isn't really a question, it's sort of a parallel. I've been working a great deal on combat traumatic brain injury.
Oh, yeah.
And your statements about sleep and the role in recovery are very pertinent to recovery from TBI, even mild TBI, where the combat soldiers have impaired sleep due to a combination of TBI, headaches, and PTSD. And actually a simple intervention to deal with impaired sleep improves their cognitive function, and most of their problems are cognitive rather than mental.
Right, it's very interesting. I think that we may be greatly undervaluing the role of sleep in our overall health, cognitive, and motor status, both normally and then also after any kind of disease or damage. So, yeah, I say that as I'm giving this talk at 4 a.m. my time, right? So I'm your role model for it. Yeah, it's interesting. I don't know of anyone who's doing TBI and sleep work, but it would be a very fruitful area. Yeah, great. I'll look forward to hearing what you find. Well, thank you very much. You feel free to contact me later if you have other questions.